Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Unit 5 Biology for October 2022. Let's begin with the first question. Question 1. Mitochondria supply energy for the processes in cells. How many of these statements about the inner membranes of a mitochondrion are correct? The first one, it is folded. It has Christie. That is correct. It is the location of ATP synthesis. That is also correct. It is the site of the electron transport chain. That is also correct. It is a site for protein synthesis. That is wrong because protein synthesis occurs in the ribosomes. So there are only 3 possible correct things, and therefore C is my answer. The next part says, anaerobic respiration produces lactate as a waste product, which is the result of lactate production. Blood becomes less acidic. This is wrong. It becomes more acidic. Muscles become less fatigued. That is wrong. During lactate production, they become more fatigued. The pH of the blood increases. No, pH decreases because, of course, the hydrogen ion concentration is increasing. And D, muscles become more acidic. That is correct. So the answer is a D. Moving on. In part B, describe how chemiosmosis is involved in the synthesis of ATP. Every time they ask about chemiosmosis, you have to talk about the electron transport chain, the hydrogen ions being pumped into the intermembrane space, creating an electrochemical gradient, and then those protons returning through ATP synthase in order for ATP to be produced. So I said electrons are moved through the ETC. This is the electron transport chain, and energy from this is used to pump the protons into the intermembrane space. This creates an electrochemical gradient. Then the protons move back into the matrix through ATP synthase. Then ADP and PI combine to form ATP. The next part says, a culture containing respiring cells was supplied with radioactive oxygen molecules, which will be the first substance to contain radioactive oxygen atoms. In the process of respiration, oxygen is going to be the final electron acceptor, and that process leads to formation of water. So ATP, no. Carbon dioxide, no. And then NAD, no. It's only water that is going to contain the oxygen that is going to be visualized, or the oxygen that is going to be radioactive. So this brings us to the end of question 1. Let's continue to question 2. Question 2. The giraffe is the tallest mammal and has a long neck. The photograph shows a giraffe. They ask, which structure joins the bones of the neck together? Ligaments join bones to bones, and therefore my answer is a B. Then the next part says, which structure joins a muscle to a bone? Of course, that is going to be tendons, and the answer here is a D. Which chemical group contains actin and myosin? Actin and myosin are protein in nature, so the answer should be a D. They are neither A nor B nor C, so the answer is a D. Moving on. In part B, giraffes must bend down to drink water. We can see that is the knee joint. They say explain how the knee joint can be held steady in this position as the giraffe drinks. Now to be held steady, of course, antagonistic muscles are going to be working. So I said as the giraffe bends, a pair of antagonistic muscles work around the joint. They could be extensors and flexors. The muscles will contract with equal strength. Also, the ligaments hold the bones in position. So during the bending of the knee, ligaments are going to be there to hold the different bones at a specific joint in position. And then muscles are also going to give support to the bones that are held in specific areas. So those muscles are going to be antagonistic. And for this bending to be supported, the muscles have to contract with equal strength. So down here, describe the role of ATP in the sliding filament theory of muscle contraction. ATP will be hydrolyzed. Energy will be provided for the myosin head to be moved from one position to the next in order for muscular contraction to occur. So I said ATP is hydrolyzed to provide energy to allow the myosin head to change position and bind onto the actin and thus form the actomyosin bridge. Also, ATP binds onto the myosin head, and this allows the myosin to be released from its binding site on actin. So this brings us to the end of question 2. Let's continue to question 3. Question 3. The diagram shows a relay neuron. This is the example of a relay neuron. They say complete the table by filling the names of each labeled part. Part L, this could be dendrite, but they could allow dendron. M is going to be the cell body, of course. And then N is going to be the axon. Then we have the O, which is going to be the presynaptic node. Moving on. Here they say lidocaine is a drug that is used as a local anesthetic. Lidocaine works by, if it's a local anesthetic, it's going to prevent formation of an impulse in the sensory neuron. So it's going to be blocking or inhibiting sodium ion channels, and my answer was a D. Moving on. In part B, the photograph shows the nerve with an outer layer of connective tissue. So that is the outer layer. They say state the type of microscope used to provide this image. We know this is going to be an electron microscope, but look at the magnification. It's quite low to be a transmission electron microscope. So this is going to be a scanning electron microscope. And another thing is this image is 3D, so it's only scanning electron microscopes that can take 3D images, at least among the electron microscopes. So the answer was scanning electron microscope. So moving down here, they say calculate the thickness of the connective tissue layer between the points X and Y and give your answer in micrometers. So we know magnification is equal to image size divided by the actual size. All I needed to do is to measure that and then convert it to the specific units. Because I want my answer in micrometers, I could have converted what I measured into micrometers. Now when I measured, I had 11000 micrometer. Of course, you can measure using a ruler and then convert the units from centimeters to micrometers. So my magnification is equal to the image size divided by the actual size. And making the actual size the subject, I got that divided by that, which gave me 3.33. I left my answer in 3 significant figures, so it is 3.33 micrometers. Now, if you had worked in centimeters, you would have got this answer in centimeters, and then you had to convert it to micrometers because the answer was required in micrometers. Moving on. Here they say, the nerve shown in the photograph contains many myelinated neurons. Explain why a myelinated neuron conducts an impulse faster than non-myelinated neuron of the same diameter. Of course, this question is about the idea that myelination allows conduction to only occur at the nodes of Ranvier, while if there is no myelin, then conduction is going to occur all over the whole axon, and that is going to slow down the impulse transmission. So I said, the myelin sheath on myelinated neurons acts as an insulator, and conduction will only occur at the gaps between the myelin sheaths. These gaps are called the nodes of Ranvier. So since depolarization only occurs at the nodes of Ranvier, it's going to be faster conduction occurs. So impulses appear to jump from one node of Ranvier to the next. So this brings us to the end of question 3. Let's continue to question 4. Question 4. Animal species have different resting heart rates. The table shows the resting heart rate in mean life expectancy for some animal species. So we have the different species. We can see elephants to hummingbirds. We see the resting heart rate is increasing as we move from the bigger to the small one. And then the mean life expectancy is actually decreasing. Generally, there is a decrease. The mean mass, we can see there is a general decrease from the biggest animal to the smallest. And then we can see the mean resting metabolic rate is also kind of decreasing. There is an overall decrease. And actually, you can see overall increase as you go down, overall decrease as you go down, overall decrease, and overall decrease. So here they say, describe the relationship between resting heart rate and the data shown in the table. So this is going to be resting heart rate. It means I'm going to consider my results in relation to that. So we can see as the resting heart rate increases, the mean life expectancy generally decreases or it lowers. And as the resting heart rate increases, we see there is going to be a decrease in that and a decrease in that. So I say generally, the higher the resting heart rate, the lower the life expectancy. The higher the resting heart rate, the lower the mean mass, and the higher the resting heart rate, the lower the mean resting metabolic rate. So those were my 3 possible answers. And down here they say, calculate the volume of oxygen produced each hour by a resting elephant with a mass of 4,500 kilograms and give your answer in standard form. So when we go to the elephant, we can see the mean mass is 5780. So I say the 5780 kilogram mass elephant produces 1,400 decimeters cubed of oxygen. You can see that to that. So it means a 4500 kilogram elephant produces that divided by that times that. We're using proportions, or you'd say specific ratios. So my answer was 1089.965, which I rounded off because actually they want my answer in standard form. So I said moving 1, 2, 3. So it's going to be 1.09 times 10 power 3. The answer is in 3 significant figures, but written in standard form. So this was my answer. Moving on. Here they say the photograph shows a cheetah. The heart rate of a cheetah increases from 120 beats per minute to 250 beats per minute during a chase. It can reach speeds of 100 kilometers per hour. Explain how the heart rate of a cheetah can be increased during a chase. This is basically explaining the whole detail of increase in heart rate during exercise. So the key thing here is, during exercise, carbon dioxide is produced that decreases the pH. Then chemoreceptors are going to detect that and send impulses to the cardiovascular center in the medulla. And then impulses are going to be sent through the sympathetic nerve to stimulate the sinoatrial node in order to increase the waves of excitation. So I said, during the chase, increase in carbon dioxide concentration causes lowering of blood pH, and this is detected by chemoreceptors in the blood vessels. These receptors send impulses through sensory neurons to the cardiovascular control center in the medulla. Then, impulses are sent via the sympathetic nervous system to the sinoatrial node so that more electrical excitation is sent to the AVN, the band of His, and then the Purkinje tissues in order to cause more stimulation of the whole heart. And then this frequency of electrical excitation causes an increase in the heart rate. So this could earn you 4 marks for that question. Moving on. Suggest why a cheetah can maintain this heart rate and speed for only a short period of time. Every time increase in muscle action occurs, there is going to be a lot of respiration, so a lot of oxygen is required if oxidative phosphorylation is to be continued. So because there is less oxygen entering, there is going to be lactic acid accumulation and the muscles are going to fatigue. So I said, during intense exercise like running, anaerobic respiration occurs in the muscle cells, and this leads to the buildup of lactic acid. Also, during sprinting, cheetahs use fast-twitch muscle fibers, and these fatigue quickly due to insufficient ATP for muscle contraction. So this brings us to the end of question 4. Let's continue to question 5. Question 5. The photograph shows a camel. Camels are an important livestock species. They are adapted to living in hot, dry environments. How many of the following adaptations enable a camel to reduce its water loss? 1 is production of very concentrated urine. That is good. Higher glomerular filtrate rate. This would cause increase in water loss, so that is out. Long loop of Henle. That is right. And reduced release of ADH. That would be wrong because release of ADH allows for more reabsorption of water, and that would be increasing, not decreasing. So that and that are going to be allowed. So the answer here is 2 because only 2 possible things were correct for the question that is asked. Moving on. In part B, the table shows the water use of 5 species of animal. So we have a buffalo, a camel, we have goat, sheep, and zebu. So here we have the water in 10 meter cube per kilogram per day and the mean mass of each. They say calculate the difference in the volume of water used in a day by a buffalo and a camel and give your answer in decimeter cube to 2 significant figures. So I began by looking at the volume. Remember, this is mean mass in kilograms, but this is per kilogram. So if you want to know the total amount of water, it's going to be that times that. So volume of water by a buffalo is going to be that times that, which gave us that, centimeters cubed. And then the volume of water for the camo is going to be that times that, which gave us 48450 centimeters cubed. Now, the difference in the amount of water used up is going to be that minus that, which gave us this. But the answer is supposed to be given in decimeters cubed and not centimeters cubed. So to convert centimeters cubed to decimeters cubed, you have to divide by 1000. So I got 56.1 decimeters cubed. And to convert these to two significant figures, it became 56 decimeters cubed. And that was my answer. Moving on. Here they say, suggest two reasons why there is no correlation between water use and mean mass of the species given in the table. If you go back here, we can see the buffalo is lighter, but the camo is bigger. So we would expect the camo to use more water in comparison to the buffalo. However, because these two organisms live in different habitats, they have different adaptations to water use that enable them to survive. For example, the camo can use metabolic water because it has a lot of fat deposits. It doesn't have to consume a lot of water in order to survive. So here I said, a camo uses less water than a buffalo, even if the camo is heavier. The camo has a hump with fat deposits, and this can be respired to produce metabolic water. Also, the organisms live in different habitats, so they are adapted differently to water availability. Part C. The kangaroo rat is another animal that lives in a hot, dry environment. In an investigation, kangaroo rats were given either fresh water, seawater, or no water. The graph shows the mean body mass of kangaroo rats, which were fed on a diet of soybeans over a 16-day period. So from this graph, we can see vertically, we have the mean mass, and horizontally, we have the time in days. Those that were fed on no water had their mean mass decrease. Those that were fed on seawater, those that were fed on fresh water, there was a tiny decrease, and then an increase, and then we see stability being maintained. So, of course, we see whether they were given seawater or fresh water, there is a similar trend, although fresh water had more increase in mass. So, let's move on. Here they say, determine the effect of the type of water given on the mean mass of the kangaroo rat in this investigation, and they want you to use the information in the graph to support your answer. This is similar to what I've already explained while looking at the graph. We see overall there is an increase in the mass of the kangaroo rats given both types of water, meaning those that were given fresh water and seawater. If we go back here, there is a general increase, you can see. Overall, I'm not saying everything, but overall there is a general increase, while there is a decrease all the way for that, because there was no water. Fresh water caused greater increase in mean mass than seawater, and kangaroo rats given fresh water had an increase in mean mass of 1 gram for 16 days, while rats given seawater had an increase in mean mass of 0.5 grams after 16 days. Usually, you talk about the general trend, the tiny trends, and then give some data from the graph, which is going to be like, maybe it could be percentages, maybe it could be a change in mass like you can see here. At least there should be quantifiable data from the graph that you can write down in order to earn the third point. In part D, they say the kangaroo rat's kidneys play an important role in the process of conserving water. Explain how the nephrons, meaning kidney tubules, in the kidneys of kangaroo rats are able to produce very concentrated urine. From your textbook, you study about nephrons with longer loops of Henle, and the purpose of the longer loops is to allow for more reabsorption in order to concentrate the urine. So I say they contain longer loops of Henle, and their medullas have a higher iron concentration. So these adaptations ensure that these kidneys reabsorb large amounts of water, and this can lead to more concentrated urine. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Plants respond to external stimuli. Phytochrome molecules are found in plants. Phytochrome exists in two forms. There is PR, which is 660, and PFR, which is 730. Which row shows the action of these two forms of phytochrome? So remember, phytochrome PR absorbs red light, and this one here will absorb far red light. So absorbs far red light is going to be PFR, and breaks down in the dark, that is also going to be PFR. So the answer is going to be an A. Moving on. In part B, Auxins like IAA or indole acetic acid are plant growth substances. The diagram shows auxin molecules producing a phototrophic response in a shoot. So here they say, explain the role of auxins in this phototropic response and use the information in the diagram to support your answer. When we look at this, we can see that when the sun or when the light is directly above the shoot, there is even distribution of auxin molecules. But if light is directional or unidirectional, we see that the auxins are going to accumulate at the shaded area and they will cause elongation in that area. Auxins accumulate at the shaded side of a plant shoot, and this causes elongation of the cells on the shaded side. This causes the shoots to grow towards light. And finally, the auxins cause the transport of hydrogen ions into the cell wall. And again, this is explaining the mechanisms of action. So this decreases the pH, causing enzymes to be activated. These enzymes break down the hydrogen bonds in the microfibules, weakening the cell wall to allow expansion to occur. And again, the fourth point is about the mechanism of action of auxins causing this expansion to be allowed. Moving on. Here they say, seeds contain starch. The starch is used to supply energy when the seeds germinate. Gibberellins are a group of naturally occurring growth substances, and gibberellins regulate starch hydrolysis in seeds. Explain how gibberellins can produce this effect on the seed. Of course, because they regulate, they will bind onto receptors on the surface of cells, causing the formation of a second messenger inside the cell, and this second messenger will act as a transcription factor for the amylase gene to be activated. Then transcription as well as translation will occur so that the protein, which is amylase, is produced, and then the amylase will break down starch into maltose. Moving on. In part D, indole acetic acid, IAA, is a natural plant auxin, then NAA is a synthetic auxin. The effects of these two auxins on growth of chlorella was investigated. Chlorella is a single-celled photosynthetic organism. The graphs show the effect of different concentrations of the auxins on the growth of chlorella. These are two different auxins. One is IAA and the other is NAA. On the vertical axis, we have the number of cells times 10 power 6 per centimeter cubed, and on the horizontal axis, we have time. We can see that for IAA, the control group had a lower number of cells per centimeter cubed, and then for the others, the higher the concentration, the lower the number of cells, because we can see 10 micromole had lower, yet 1 micromole had higher, and 0.1 had a higher number. So we can see the higher the concentration, the lower the mean number of cells, or the lower the number of cells per centimeter cubed. And we can see for NAA, the control had a lower number of cells per centimeter cubed, but also we can see 1 micromole per decimeter cubed had the highest number, and 10 micromole had a lower number, yet 0.1 had a lower number than the 10 micromole. So these results are not as predictable as we can see with IAA. So here they say, comment on the effect of these auxins on the growth of chlorella. So I said both IAA and NAA caused increase in the number of cells compared to the control. And again, I hope you can see that for all the controls were lower than those that had the different concentrations of the growth factors. IAA led to more growth compared to NAA. And for IAA, the most effective concentration for growth was 0.1 micromole per decimeter cubed, while for NAA, the most effective concentration for cell growth was 1 micromole per decimeter cubed. So in conclusion, IAA is better than NAA because with 0.1 mole per decimeter cubed of IAA, the cell concentration was about 44 times 10 power 6 cells per centimeter cubed, while at 1 micromole per decimeter cubed NAA, the cell concentration was 25.8 times 10 power 6 cells per centimeter cubed. So we can see you use a lower concentration of IAA, you get a higher number of cells in comparison to NAA. That is why IAA is better than NAA. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Animals and plants can be genetically modified. Recombinant human erythropoietin, RHE, is a protein drug that can be given to patients with low hemoglobin concentrations. Sheep have been genetically modified to produce RHE in their milk. Explain how a sheep could be genetically modified to produce RHE in its milk. So for genetic modification, of course, we have to identify the gene and then find ways of isolating it, use restriction endonucleases, then we can transfer that specific gene into a suitable vector and then we insert that into a specific cell. So I said, identify the human erythropoietin gene and isolate it, use restriction endonucleases to cut the DNA sequence for that gene, and then insert the gene into a suitable vector. It could be a plasmid or a virus, and then insert the vector into the sheep udder cells because the aim of this is to be produced along with the milk, so it has to be in udder cells. You could also say insert this gene into a zygote so that the zygote's udder cells in the future will be able to produce milk because the gene will be already introduced there. Moving on. In a clinical study, 4,382 patients were treated with RHE. They showed a mean increase in blood hemoglobin from 80 gram per decimeter cubed to 142 gram per decimeter cubed. They wanted to calculate the percentage increase in the mean blood hemoglobin concentration as a result of RHE treatment. So the increase is going to be 142 minus 80, and this gives us 62 gram per decimeter cubed. So the percentage increase should be that divided by the original, which was 80, times 100, so we get 77.5%. Moving on. Here they say some patients receiving RHE develop anti-RHE antibodies. Describe how these antibodies may be produced. The RHE acts as an antigen, and so macrophages engulf the antigen and carry out antigen presentation. Then the T helper cells will become activated due to that, and this will cause the activation of B cells. Then the B cells will differentiate into B plasma cells, and these B plasma cells are going to produce anti-RHE antibodies, and then these are the antibodies against the RHE. And again, maybe I should put it here, anti-RHE antibodies. Moving on. In part B, advances in genetic engineering have allowed for precise control of genetic changes introduced into plants. The table describes some examples of the use of genetic modification in plants. For herbicide tolerance, we have soybeans, the glycophosphate herbicide tolerance using a gene from tumefactions. And then insect resistance is present in corn, and again, resistance to European corn borer using a gene from B. thuringiensis. So they say the graph shows the increase in the use of genetically modified crops in the United States from 1996 to 2017. When we observe the graph, on the vertical axis we see percentage of area planted that is genetically modified crop. On the horizontal axis we have the time in years. Initially, there was a high increase in area where soybeans were planted in comparison to corn, but from the year about 2014 we can see the area where corn is planted increased greatly. Moving on. So here they say discuss the risks and benefits of genetic modification in valuable agriculture plants and use all the data and your own knowledge to support your answer. So of course, the benefits are it increases the yield. Many people are going to be fed. People are going to have a balanced diet because now we can genetically modify the crops in order to introduce specific nutrients that are required to give a balanced diet. So malnutrition will be dealt with. Problems are going to be it's going to take up land, and then those pesticides, there is a possibility of horizontal gene transfer. So I said, modification allows for more crops to become resistant to herbicides and insects. Corn will no longer be eaten by the corn borer, and soybeans will not be killed by the herbicide. As a result, more corn and soy was grown from below 25% in 1996 to above 75% in 2017. This increased the yield, providing more food for the increasing population. Also, with genetic modification, the nutrient content in the plants can be improved, producing crops with balanced nutrients to deal with food deficiency, disease, and malnutrition. However, there is increased need for land for agriculture, so forests may be cleared. Also, there is a chance of monoculture, so monoculture could result in farmers growing only the GM crops, leading to low genetic diversity. Also, there are unknown consequences of foreign DNA in humans. So some people are worried about that. So there is also horizontal gene transfer that could occur in nature. So this brings us to the end of question 7. Let's continue to question 8. To answer question 8, I want us to first look at the scientific article. So this is the article. If you can look back in other files, I went through this article. So you can look through it. I'm just going to show you how it is, and then we can begin to answer the questions. So we can see this was about basically nerve impulses and then generation of impulses, basically. So we can see that. Channels and so on. Receptors. And that is the end of the article. So let's go to the questions of question 8. So question 8. The scientific document you have studied is adapted from an article, How We Perceive the World, by Julius D. and Pataputian A. in 2021. Use the information from the scientific document and your own knowledge to answer the following questions. In part A, describe how light is detected by the rod cells in the eyes. This is paragraph 1. And again, this is easy, even if you do not go back to read. This is from your notes. We know when light is absorbed by the rod cells, bleaching occurs, and then the cis-retinol is converted into trans-retinol. The opsin and retinol are produced. Then the sodium ion channels in the membrane close, and then the membrane becomes hyperpolarized. So moving on. In part B, suggest how the genes expressed in a sensory neuron could be identified. Identification of expressed genes can be done using microarray. So in this case, we will extract the messenger RNA from the neurons and then use reverse transcriptase to form cDNA. Then we use microarray with fluorescent dyes and a probe, and then compare unstimulated and stimulated neurons using bioinformatics and analyze the data. Or you could extract messenger RNA and use reverse transcriptase to produce cDNA, and then amplify using PCR, and then use gel electrophoresis and compare the specific bands. So these two approaches are acceptable. Moving on. Here they say, explain how capsaicin could activate nerve cells, causing a pain sensation in paragraph 4. From paragraph 4, we can see, of course, this is going to bind onto receptors on the surface of cells, and this can cause the opening of ion channels in the neurons. Depolarization can result, and if a threshold is reached, then an action potential is going to be generated, and then relay neurons then transmit the impulse to the brain for pain to be perceived. In part D, explain how changes in ion channels can result in nerve impulses in the nervous system. When stimulation occurs, ion channels will change their shape, and this can lead to opening of the channels. So sodium ions will flow into the neuron, and then depolarization will occur, and an action potential is going to be generated in the specific neuron. Moving on. In part E, explain how mechanical stimuli, example pressure, are converted into electrical signals in the cells. Again, this is the same story. Pressure changes. So basically, pressure changes is going to cause the opening of the ion channels. Either ions are going to flow in either sodium or calcium, and then depolarization will occur. So I say the stimuli, example pressure, can change the shape of the membrane. The ion channels can open. Then there is influx of ions. This could be sodium or calcium occurring. And then depolarization can occur, and an action potential can be generated. In part F, explain what is meant by the phrase, based on its similarity to piezo1, a second ion channel was formed piezo2 in paragraph 7. I said the structure of piezo2 could be similar to that of piezo1, or both are activated by pressure changes, or both could be transcribed from similar base sequence, so DNA or amino acid sequence analysis could have been done. So any two of those three could earn you the 2 marks. And lastly, they say suggest the role of TRPV1 in maintaining core body temperature. TRPV1 was sensitive to temperature changes, so I say TRPV1, a channel sensitive to the temperature of the blood, meaning this is the core body temperature. So they send impulses to the central nervous system mainly the hypothalamus, and then the hypothalamus participates in triggering negative feedback to achieve homeostasis. So this brings us to the end of question 8, as well as to the end of this whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.